Alright, welcome to our video this week. We are going to start off and we're going to talk this week all about senses. So, start going through some senses here. In our eyes we have photoreceptors. That detect light. In our ear we have mechanoreceptors that detect vibration. And sound through use of mechanics. We'll talk about that a little bit later, and you'll be able to see. And then we use chemoreceptors in our nose and in our mouth to detect, so they detect chemicals for smell and taste. Okay, we're first going to talk about the chemoreceptors. So if we draw a picture of a nose, so nose, when we smell something, alright, that's supposed to be a pie. It doesn't look like a pie, but so we have these different parts of our nose. So up at the top, we have what's called an olfactory bulb. Underneath the olfactory bulb, we have a little section of bone. And underneath, we have our olfactory epithelium. And up here we have stromatous stars. These are mitral cells. And they go back and over to the brain. And then we have these olfactory neurons that are running through here. And there's a lot more than this. I'll just draw just a couple. Synapse with those mitral cells. And so we have these particles of chemical that'll come up and we'll be able to smell them. And so the way this works, I'll draw a closer picture of the, oh, I never labeled these ones. 3. Oh. Neurons. So, these are responsible for smell. So I'm just gonna write. So I'll draw a bigger picture of this. We have these channels here. We have a section here called adenylate cyclase. And so once we have one of these particles come up and bind, what it does is it activates, it sends forth a signal and activates adenylate cyclase. Adenylate cyclase releases cyclic AMP. And what that does is that actually opens a channel that's right here. This is a non-specific cation channel. And what happens in this olfactory neuron is we have a lot of chloride. It's going to leave the cell, and what's going to rush in is sodium and calcium. And what's that's going to do is create a gradient potential, making us have an action potential further down the line, which, if I remember correctly, this actually doesn't create a action potential. Instead, it creates a sorry, I just lost my train of thought. A graded potential. Many graded potentials, which eventually will reach threshold and have an action potential, but it's through a different process that allows us to do that. So we mainly just focus and call it just a graded potential. Potential. Okay. Now we're going to talk about taste. So, I'm going to draw part of a tongue here. This tongue, we're going to have little sections. Right at the bottom of these, we have what people refer to as our taste buds. And a typical person has about 125 taste buds. Not all of these, some of them will be sensory and send messages to the brain, but not all of them. A lot of them are mainly for support. The way this works is down here at the bottom. This will synapse. So I'll just draw it a little bit bigger, just as we've talked about before. We've got these receptors at the bottom. This has our snare proteins that are trying to grab on. These have neuroreceptors inside them that are trying to send signals to the brain for these receptors. So this will also do graded potential. I'm sorry about this. This actually does create an action potential. I got that backwards. Action potential. But taste actually just does these graded potentials. And so we have these different forms of taste. We have salty. And this is, the chemical responsible for this is sodium. As you think about salt, it is sodium chloride. Sweet is carbohydrates. Bitter, caused by calcium. Sour is hydrogen. And umami is glutamate. And so, the process is happening. So these three will all be the same. So if you just think about this square being changed to these different processes, so first we're going to start here on sweet, bitter, and so this actually uses a ligand gated channel. And as the ligand comes in, what it does is it sends a signal to the endoplasmic reticulum in here that is filled with calcium. And it sends that signal right here to this calcium channel and releases the calcium into the cell. And what that does is that creates a graded potential, eventually allows us to send our neurotransmitters down. Salt uses sodium to come in through a channel here. What that channel does is it activates a voltage gated calcium channel. And that opens and that flows in, and as that flows in, it creates a graded potential once again and is able to send those neurotransmitters down. Sour does a very similar process where it takes the hydrogen in through this channel, and this also activates the voltage gated calcium channel. Send calcium in, which as well makes a greater potential enough to send those neurotransmitters down and create a signal right there. All right, so now we are going to talk about the eyeball, vision. So as we already mentioned, the eyes use photoreceptors to detect light. So how they do that and how they focus on it is very interesting, and it's all mainly due to anatomy. So first we need to draw an eye here. On the eye, you have a part that comes out. This is called the cornea. Cornea comes from a derivative of a word that means hard. And then inside we have this muscle group all around. And specifically right here, these are called the ciliary muscles. And then we have these coming right here. These are called suspensory ligaments. And what they do is they hold this part of the eye. This part is called the lens. And so these are both convex types of mirrors, as you'll learn about it in physics. Essentially what it's going to do is it's going to curve the light. So we draw a convex. What it's going to do is it's going to curve the light back into a certain focal point. And so we do have a focal point in the back of the eye. This is called the macula. And then in the center we have the fovea. So, and then the fovea. So in both of these have a big excess of these photoreceptors. I'll talk about these different types of photoreceptors in a second. And then right here we have, this is called the natural blind spot. This is the optic nerve. The optic nerve is where these synapses run out of the eye and where there's that opportunity for it to connect to the brain. So right now I'm going to actually talk a little bit about these ciliary muscles and the lens. So as we have a focal point up close, say that we're reading a book. We have two different close up and then far away. I don't know, like a street sign or something. So this is farther, this is closer up. Let me redraw that. Our focal point. It goes right to the edges of the eye. And then it's got to come back. And most of the light hits the fovea. But for farther out, it doesn't have as sharp of an angle right there. So close up has a really sharp one, and far away has a more obtuse angle. So in this same sense, the ciliary muscles, they have to contract for close vision, and they relax for distance. All right, so now on to these photoreceptors. So we have two main kinds of photoreceptors. We have rods and cones. And rods are black and white, and cones are colored. There are more rods in the eye than there are cones, but cones are uh, your higher definition vision. That's how you see things in greater detail. This is more just general vision. And this is very sensitive to light. They call it photo bleaching. Like, say in the morning when you get up to go to work, you flip on the lights and you instantly have that flash of light in your eyes saying this is way too bright. That's all due to your cones. And this is, I guess, this has more affinity for light. Okay. And so now we're going to kind of talk about this mechanism of how light actually works. And it's going to take a little bit for me to draw it, but something I should mention here, and I'll just show it here. So within the fovea and in the whole back of the eye, this is called the retina. Retina is all the back of the eye. Also, something I forgot to mention is we have different chambers of the eye. This is the anterior chamber. This has aqueous humor. And here in the back, this has vitreous humor. So it's all this liquid type of stuff. And so with the retina, so we'll have, these are blood vessels. When you go to your optometrist, they'll take a picture of the back of your eye, and usually you can see the fovea, the optic nerve, and then you can see a bunch of blood vessels. Underneath those blood vessels, we have this layer, and this is called the outer pigment of the retina. And then next, so we have these photoreceptors that we were talking about. Here's my best guess of what a rod would look like. Here's a cone. And then after that, so we have rods, cones. Next we have this layer right here. And these are the bipolar cells. They're kind of in between. And then lastly here, we have the ganglion cells. I'm going to draw them like this. And then these ganglion cells, they'll actually attach here, and they'll all meet up at a certain point. And all of these together will leave the eyeball, and these all make up the optic nerve. Okay, and so what we're going to do is we're going to zoom in on one of these cells here, one of these photoreceptors. We're going to talk a little bit about what is going on here. So, first off, we have a couple things that are important to know about. So, when the light comes in, so our first step is the light. Light will hit this molecule here. What this is called is 11-cis retinol. And 11-cis retinol will go through a conformational change. It'll actually turn to 11-trans retinol when light hits it. What this does, when it changes, it actually activates something called opsin. And opsin, so this opsin will activate something called transducin. And what transducin does is it makes conformational shifts, and it will send an alpha molecule over, which will activate phosphodiesterase. And phosphodiesterase has a direct impact on cyclic GMP. And so, cyclic GMP, so we actually have a glycic protein channel, and this will go and inactivate it. Which what this does is it takes GTP and changes it to cyclic GMP. What it actually does is it takes it and turns it into just GMP without that. So the major impact on that is here. We have these channels throughout the cell that normally let in sodium and calcium. These are closed because we do not. This requires cyclic GMP. So these are closed. What this causes is hyperpolarization. And in effect, this causes and IPSP. So as we come down here to our photoreceptor, we have all of these neuroreceptors that do not have the chance to synapse. We also have these voltage gated calcium channels that are still not open because we don't have this. So typically, glutamate would be present here, but because this did not synapse, we do not have the chance to do that. But what the effect is on this other cell, we have a receptor here called M-glutarate G. There's a receptor right here for glutamate. But the effect is, when this is active, it actually inactivates another channel called TRPM1, which is a voltage-gated calcium channel. So when this is active, this is inactive. But in this case, we have an inactive receptor and an active voltage-gated calcium channel. So as our calcium comes in, we grow a graded potential, which, with this separation of charge, which helps us to reach membrane potential and be able to have a synapse. So this allows us to have a synapse right here on this bipolar cell. So right here, if we remember, we have our G and our cell R TRPM1, and calcium comes in. And what this does is it sends an action potential down the cell, releasing an action potential, which is received, and it sends it down the cell. And so, alright, so the synapse happens and it sends signals to the optic nerve. We have special situations in the other way. We have this, so we have different rods here. And this is called the center, and these are surrounding. And they have different mechanisms. So we have our rod, and we have our bipolar cells. And this is called actually a horizontal cell. So I could draw them here, these horizontal cells. All right. Off of this. To say if that's that. Horizontal, boxes. And this is our ganglion cell. Okay, and so we have different kinds here. So we have a on center and an off surrounding. The way this works is so they are completely opposite. The on center desires to have light on it to create a synapse, and the surrounding does not. So say we shine light on 2/3 of this. So this part gets light, and this does not. And we kind of have a rule we follow here. And it's backwards of what you'd think. Dark light. Dark equals more glutamate. So what this is responsible for, this is responsible for 60% of the synapse, and then this is 20, and this is 20. So if we have more light, we're going to produce less glutamate. And you're thinking, how does that happen? So if we think back to this first example, the glutamate actually makes the synapse not happen. Makes us have less action potentials. So it's the same concept here as that light, as the light hits, it works through and we send neuroreceptors down. Well, right here. So it does not produce glutamate. That light's on it. Not produced, but it still sends a signal down. And as this one has the light shown on it too, this actually does not produce glutamate. Which these have a little bit different of a system with these horizontal cells. What they do, so this one would produce glutamate. But as it produces glutamate, it has this thing called AMPA. What AMPA does is it senses this glutamate and it inhibits it. So it sends less glutamate. So because we have 60%, we have 6 action potentials happening here and 2 action potentials happening here. So it has no light on this one. I know I drew light on it before, but don't mind, my light disappeared. So this would cause us to have 8 action potentials. And so I actually think that we are just about done here. It is a little bit different with the off-center and the on. So we have off-center and on surrounding. It is a little bit different. It uses some different enzymes. I'm sorry, I actually messed this up. This is GABA. This is GABA to inhibit it. And so if we have off surrounding, this desires to be off. And it works in the same manner that we had on the off surrounding before. So it produces this. But then this actually isn't glutamate. This is called AMPA. And AMPA just produces that action potential and doesn't inhibit it. These still work the same way that GABA So as light does not hit it, it produces the glutamate, and GABA will inhibit it. And so if we have light hitting half of this, this will not produce an extra potential, because what it will do is that light won't hit there and it will not produce AMPA. Light will hit here and it will produce that glutamate, and the glutamate is then inhibited by GABA, stopped right there, and we're able to have an extra potential. So that is all we have to talk about for. All right, so now we are going to be talking about our sense of hearing. So hearing is a pretty interesting one. It takes sound waves, turns it into mechanical waves, uses more of like a water wave, and ends up sending it into an electrical signal back to the brain. So first off, what I'd like to draw out though is how this works. So we have your ear. Here's your external auditory medius. So when you hear something, send the signal down. So this is our outer part of our ear. So it's responsible for the vibration signal. And then we hit the middle part of our ear. We have three parts of this. We have the malleus, the incus, and we have the stapes. What the stapes does is it moves. Well, this whole thing moves. It's on an axis, and it moves back and forth and sends this signal right to, this is the oval window of what is called the cochlea. And the cochlea comes up like this. Kind of looks like this spiral. If we were to cut this in half, we'd be able to look at it like this. I'm going to try this. Okay, there's our oval window. Here's our stapes. We're going to divide it in thirds here, and there, and there. And so we'll label these. This is the scala vestibule, scala media, and scala tympani. So both the scala vestibule and the scala tympani have what is called perilymph. And this is the liquid that's in here. Their continents are pretty similar. The scala media, what it has is endolymph. And then there's a section called that's right here. This is called the tectoral membrane. This is connected. So, another thing, so when this sits and taps, the layer right here, that is long, and then it goes shorter. So when this taps, what it does is it sends a signal through the scalar vestibule. It comes down, or eventually hits here and sends a signal over. But these hair-like projections, they're actually just called hair cells. That come over, and as they move, they touch this tectoral membrane. I'll accept this one. This is the internal. These are the external. The difference between these is the internal actually goes to the brain and is mainly responsible for what we hear. The external are mainly for regulation, and we'll talk a little bit about that. They also can send some signals to the brain, but mainly are for regulation. So I'm going to draw one of these hair cells. And something that's important about the scala media and the endolymph is it is high in potassium. And these structures are like this. They have these gated potassium channels that are on this connecting rod, and they're closed. But as we move this membrane, as it moves up and goes close to the tectoral membrane, it eventually touches it. And what happens is this moves over like that, and it opens those channels, allowing for that potassium to come in. And as it comes in, it also allows for this opening of a voltage-gated calcium channel come in. What it does is it allows for depolarization, allows us to have some action potentials and send those signals back to the brain. So this releases glutamate and sends a signal back to the brain. So the external hair cells, what they do is the way they regulate is they have these proteins on the side. This is called prestin. So what prestin can do is the brain can send a signal up, and this prestin will recognize it, and it will send potassium out of the cell, creating hyperpolarization, preventing action potentials, and in turn turning off your auditory sense. So if you think about it, say you're at a concert and it's really loud, but your friend says your name, and you hear and you recognize their voice, and your name admits all the noise. These external hair cells are the ones that are responsible for tuning out the sound, and the internal will work to hear that certain sound and be able to focus on it. So it's really cool the way that things work like this. The only thing I think I forgot to mention was a human can hear 20 hertz to 20,000 hertz. And we typically never hear close to the borders of those, but it is possible for us to hear them. So we'll go through it one more time. So we will hear a sound. It'll go through our external auditory medius, our the middle part of our ear will send a mechanical signal, and it will hit the oval window of the cochlea, sending a wave through the scala, this vestibule, through the perilymph, into the scalar media, into the endolymph, sending the signal down, which will cause the hair-like structures to touch the tectora membrane, opening those sodium-gated potassium channels. The gated potassium channels, not sodium, allowing us to have that action potential and send a signal to the brain. All right, so that's all we have to share for this video today. Thank you for staying.